All right, so for this week, you have homework number 1 due. So we'll talk about that real quick, and then you just finished the quiz. We're going over cell anatomy membrane transport. Okay. So next week we're covering tissues. All right. So week 2. All right. All right. So basically, you're going to create a Google presentation. Okay. All of you have a Google account that comes with your Panola College email address. Okay. So you'll go to create a Google presentation, and you need to share it. Set the sharing preferences to anyone with the link can view. Okay. You're going to have 4 slides. First slide is your name, assignment title, homework 1, and your class, biology 2404. Second title is going to have landmarks. Okay, that'll be the title. You'll have a picture you'll find off the internet, or you can cut one out and take a picture of it. Doesn't matter to me. And you're going to label 15 landmarks on the body on that picture. Okay. And for third slide, okay, it's body quadrants. It's the title. Another picture, you're going to label those you're going to draw the 4 in the line, and then label 2 of those 4 quadrants. Okay, the fourth slide, you're going to do relative directional anatomy. Do another picture, label 6 areas in the picture, A, B, C, D, E, and F. And you're going to write 5 sentences using the correct terminology. So you can say, example, area A is superior to area F. Okay. You do all those things, you get 100. Okay. So, and then you've got to, so basically, all right. So go to my drive. Let's see. All right. So I'm going to log in. All right. So you're in the cell nucleus where your DNA is. It's where the DNA resides, right? And so you don't really need to know the inside of that. This is the nucleus. All right. Cell membrane or plasma membrane surrounds the cell. Rough endoplasmic reticulum has ribosomes. Smooth endoplasmic reticulum does not. Okay. So if you see that around the nucleus, the one that has dots on it is rough endoplasmic reticulum. The one that does not is smooth. Okay. So proteins are made at ribosomes. Okay. So we'll talk about this more in chapter 3 in lecture, but there's a important pathway, DNA, used to make RNA, which is used to make protein. Okay. It doesn't become the other thing it's used to make. And I tell you that because rough endoplasmic reticulum has ribosomes. So proteins are made there. Okay? So DNA is used to make RNA in the nucleus. It's called transcription. And translation, that RNA leaves the nucleus and comes to the ribosome to make protein. We call that translation. So, transcription, this is translation. Those proteins are then processed at the Golgi apparatus, including these little circles, we call these, or spheres, we call these vesicles. and they go to the cell membrane and get released out of the cell. Okay, via a process we call exocytosis. Exo means out, cyto means cell. Okay, so this is exocytosis. All right. Cytoplasm is just the fluid that all the organelles live in. Okay. Lysosomes, they basically break stuff down. They have enzymes. They're little chemicals that eat stuff up. They're like kind of like the cleanup, the trash service for your cell. Mitochondria, these things look kind of like pickles or jelly beans, whatever you want to call them. They make energy. And it's in the form of ATP. Okay. Centrioles, these little lines. Well, centrioles are actually like these right here. They look like, I guess I don't have a better way to describe them than like mini churros. Okay, or they kind of have the ridges, the lines on them. But they are on the ends of the cell and they help the cell spread apart when it's getting ready to divide. Okay. So, but that's an overview of the components of a cell. Okay. Now, that's a general eukaryotic cell with a nucleus like you'd find in your body. The ones with the black background in the back, those are neurons, those are nerve cells, they're still cells. So most of the structures that you'll see are still in there. It has the same structure. When we get to the nervous system, then we'll talk about the specific parts. Right? But generally speaking, whether you see one or the other, you should be able to identify parts on it. Okay. All right. Now there are also cellular extensions, things that come off the cell. So extensions mean things that are on the outside, right? So they're extensions of the cell. We have cilia, which are unlike, I mean, they're all tiny, but they're like little hairs. Okay. And they can kind of help with movement. We also have microvilli, which are much shorter. And these typically are help with absorption. Because they increase surface area. Okay, whereas these are more for movement. We have moving things out like in your nose. You want to move all the bad stuff that's in your nasal passage. Okay. And another extension, all right? This is a sperm cell. This extension is called a flagellum. Singular flagellum, plural is flagella. Okay, and that's also for movement. All right. Okay, cell membrane, plasma membrane. So there are things on the surface that you need to know. All right. All right. So this is the extracellular space outside the cell. intracellular space on the inside of the cell. So one thing you should notice right away is that there are two layers to this membrane. So it's a phospholipid bilayer. So like this is a layer, this is a layer. Okay. These little things with the little two lines like that, we call those phospholipids. Okay. It's made up of phospholipids, and there's two layers, so we call it a phospholipid bilayer. Because bi is two, right? Like bicycle, that's two wheels. All right, the heads, the little circles, we call those polar. We're going to talk about chemistry today in lab. It just basically means they have a charge. Okay. Now, the tails are nonpolar, so they don't like things that have charge. And water has charge, which is a lot of this is water in this space. So the parts that don't like water get on the inside of the bilayer. Parts that like water get on the outside. All right, inside the membrane, you've got some cholesterol, and give you some fattiness to your membrane, some flexibility. You've got proteins that are embedded. Some of them have pores, so chemicals can go through. Some of them are channels. Some of them are receptors. And so when things touch the proteins, it causes some chain reaction inside or outside the cell. You also have carbohydrates and sugars attached to your plasma membrane. Okay? But yeah, so if you have the line all the way through the protein, it's typically some kind of channel. Okay? So not channel like TV channel, but channel like things can flow through it. Okay? But I guess in a sense, TV channel means content's coming through that channel, right? So, all right, we're going to do a diffusion experiment next time, but I just wanted to introduce this to you. So diffusion means movement of material from an area of high concentration to low concentration. So you see diffusion. Talking about movement, high concentration, to low concentration. All right, so put some like a sugar cube or something in some water. At the beginning, most of it's all in one area. But if you leave it over time, what happens? It spreads out, right? It evenly distributes. Okay. So now, because you've gone to the areas where there was low concentration, it spreads out. That's diffusion. Now, in the cell membrane, we also have diffusion. And diffusion could be simple. So if materials are soluble or they dissolve or they can be in fat, like the lipid part of the membrane, they can go straight through the membrane. Okay, they're okay with the middle. If they can get through the middle, they can get all the way through. Okay, that's simple diffusion across the membrane. Sometimes, like if they're not lipid soluble, they have to require protein carrier. Okay, so we call that carrier-mediated facilitated diffusion. We call it facilitated because it needs a facilitator, right? But who's the facilitator? The protein. It's also a carrier. It's carrying sugar across the membrane from an area of high concentration to an area of low concentration. Here you have channel-mediated facilitated diffusion. Still facilitated because you have a what? What is this? Huh? It's a protein. But this time it's a channel. You just go straight through. Okay? So sometimes small molecules that are not soluble in lipids, they don't like the middle of the bilayer, will go through a channel. But sometimes the ones that are larger need a carrier. Okay? So you need to know whether it's lipid soluble or not, whether it's large or small. Okay, you need to know the concentration difference. All right. Now, diffusion, again, high concentration to low concentration. But if we're talking about water specifically, it's osmosis. So osmosis is the diffusion of water. Osmosis is the diffusion of water. Osmosis is the diffusion of, thank you. If I repeat it, it's probably important, right? It's still high concentration to low concentration, it's just we're talking about water, okay? And sometimes it can go straight through the membrane, other times it goes through a protein. All right, so hypotonic, isotonic, hypertonic. All right, solutions. If the concentration inside and outside a cell or a bag is the same, we call that isotonic. Iso means same, okay? Tonic has to do with concentration. So same. Hypos, low or lower. Hyper is higher. Tonic means concentration. All right, so the concentration outside the bag in the solution is lower than the concentration inside the bag. It's a lower concentration, hypotonic solution. In water, if the molecules can't go through the membrane, but water can, water is going to go, because now there's more water, a higher concentration of water on the outside. So water's going to want to go in. So what's happening to the bag? Expands. In a hypertonic solution, the concentration of the solute or the molecules is higher outside, which means the concentration of water is going to be lower outside. So water's higher inside. Water goes out. What happens to the bag? Shrinks. All right, so what does that look like in the cell? Well, here's your red blood cells. They normally have this little biconcave district. In a hypertonic solution, all right? So in a hypertonic solution, it shrivels, okay? Just like we saw before. In a hypertonic solution, or sorry, in a hypotonic solution, it swells. Okay? Its concentration of water is higher, concentration of solute makes it lower. That's why it's hypotonic. It just means it's higher concentration of water. Water goes in, swells, and concentration is too high, bursts. And you don't want red blood cells bursting. All right. Real quick, I know we got to go, but we've got one slide. Most of what we've talked about is passive transport. Passive transport means no ATP is required. Active transport means you require energy. Okay. In the form of ATP. All right, so here we're moving sodium ions across the membrane, moving them out. Okay? But in order to do that, you need ATP. And you're going to bring potassium in. Okay? But you're going against the concentration grid. Okay? Because naturally, it's the other way around. Yeah, naturally. You're having, so you have another system that's opposing this. This is going against what would happen normally. Going against what you would normally do is going to require energy. Okay? So, because we're going essentially against the concentration. All right, so that's going to require energy. So I'm not going to get into all the details of the pump. We'll talk about that in chapter 2. But I just want you to know it's an example of active transport requires energy. All right, sodium potassium pump, active transport. All right, so what we'll do is we'll have you you can go ahead and make your way over to lecture, and you can do your exit tickets on the way on your phones.